right, hello, welcome back. For today's coronavirus report, 45,210 confirmed cases, 175 outside of China, still amazing, 1,118 deaths. These are probably big underestimates for two reasons. One, I think you never get all the cases. You can have up to a tenfold underreporting. People feel fine and they're infected, so we don't diagnose them. And secondly, I think the system is overwhelmed. So many PCR assays per day per lab, can't handle it. So in the end, there'll be more if we learn. I'm not sure we'll learn. There is word that they're not reporting a lot of the cases, especially the deaths. I don't know how to deal with that because China is the only source of data. No one else can go in there and find out. So we either believe what they tell us or not. Not much else we can do. The number of cases, so someone said to me, it doesn't matter that there's a little plateau here because it's a delay in the reporting of cases. So that could very well be. So it may not be that it's leveling off. Today, we're going to encounter transcription and RNA processing. Last time we talked about RNA synthesis of RNA viruses. Today it is RNA synthesis of DNA viruses, mostly a process that we call transcription. And here is the cast of players. That's our Baltimore scheme, of course. mRNA in the middle. And what we're going to look at today is how viruses with DNA genomes get to mRNA. And so the ones in red are the ones we're going to talk about. We have our single-stranded DNA viruses, parvoviruses. We have our gapped double-stranded DNA viruses, hepatitis B virus. We have our virus with double-stranded DNA. And we've talked so far in this course about adenoviruses, herpesviruses, polyoma and papilloma viruses. Well, they all have to make mRNA from DNA templates. And of course, retroviruses do as well. They convert their RNA genome to DNA, to double-stranded DNA, which integrates into the host and is transcribed there. So all the principles I'm going to tell you today apply to retroviruses. There are some variations which we will encounter later. People ask me all the time, do I have to remember the names of viruses? Well, I give you, 7 different viruses. I think you could remember that. You're going to remember coronavirus, right? And all these others, just write the Baltimore scheme on your cheat sheet and put the name of a virus next to it. I will, in a question, I'll try and say, poliovirus, a virus with a plus-strand RNA genome. But sometimes I forget. And so you can do two things. You could ask me during the exam. But the way it goes is the people in the middle are inhibited to ask anything because they have to come out. So you don't get that chance. Or you could write it on your cheat sheet, which takes very minimal effort. So I would do that. Or I'd remember it. So in 10 years, when a patient asks you, what strand is this virus, you can tell them without looking it up. And you look smart, and they'll come back. Medical students always ask me, why do I have to memorize the polarity? But I say, patient's going to ask you one day. They're going to say, what's the strandedness? And I ask my doctors that they don't know, because they didn't take my course. They have no idea. They barely know what a virus is. They just know how to treat it. Yes? No, it could be done in anything. Computer, handwritten, blood, whatever you want. But blood would be a health hazard, so don't do that, right? All right, so by the way, the exam's Monday, of course. I'm going to put up the answers to the practice exam this weekend, but I want you to try it first. Don't just wait for the answers and then look at them. It's not how you learn. Try it. And then look at the answers. That's why I'm delaying it. And I will put up answers to the study questions because you need to know the right answers. I mean, they're all embedded in the lectures, but in case you don't want to do that, I will put up those so you study the right things. All right, back to transcription. Here's a general principle that you have to remember. You could write this down too. In cells infected with DNA viruses, at least one protein, sometimes many, depending on the virus, is needed for DNA replication. That's the topic of next week. So to make a protein, you have to make a messenger RNA, and that's why we do transcription first. in the sequence of the replicative cycle, because every DNA virus has to make at least one protein to get DNA synthesis going, and to do that you need to transcribe DNA into mRNA. The thing here is, not all viral DNA templates are ready for transcription. Here's an example of a virus whose DNA is ready. It's a polyomavirus, and we have looked at the structure of this virus before. We're going to look at transcription of it today. We're going to look at DNA replication next time. So it's a virus you're going to hear a lot. We're going to come back to it when we talk about how viruses cause cancer, because it was a big player in that. But today, we're going to talk about its transcription. There's the particle inside of it is the DNA genome. It's a double-stranded circle as shown on the right. And this virus is unusual because in the virus particle, the virus is wrapped around histones. Maybe you can just see the little disc-like histones with the DNA wrapped around them in the particle. There's a bigger view of it here. See, the DNA is wrapped around histone proteins. That's the way our DNA is, of course. I call it chromatinized, but it's in the virus particle here. It's quite unusual. Most viruses are not like that. Now, this DNA gets into the nucleus. It's ready for transcription because it is double-stranded, and RNA polymerase, as you will see, can recognize it. So the polyomaviruses with the small circular double-stranded DNA genome, any virus with a double-stranded DNA genome is pretty much ready for mRNA synthesis. Now, before I leave this idea of having chromatin around the viral genome is really interesting, because most viral DNAs in the particle, whether it be an adenovirus or a herpesvirus or something else, is not chromatinized. It's typically, it may have other proteins around it. It's not chromatinized. However, when it gets in the nucleus, it is immediately coated with chromatin. Cells do not like to have naked DNA in the nucleus. So whatever comes in is completely chromatinized, and often that silences it. Because when you have DNA wrapped tightly around nucleosomes, it's not good for transcription. And so there are ways that viruses counter that so they get transcribed. But viruses have to obviously get unsilenced, and so there are all different ways to do that. Retroviruses integrate, and once they integrate, they're no longer silenced. But we'll talk about some of the strategies today. Or not today, another time. All right, so this is an example of a DNA. Here, this virus enters the cell. The DNA goes into the nucleus. That's where it has to replicate. And it's immediately transcribed because it's ready. But not all genomes are ready for transcription. 3 shown here have to be converted before they're transcribed. And the Adenovirus DNA on the top, it is gapped, has a protein linked to one end, it has a piece of RNA on it. That's not ready for transcription. So when that DNA comes into the nucleus, the cell sees it. Now, so the cell nucleus, when it sees DNA, if it's broken, it fixes it, because it really can't distinguish between viral and its own DNA. So this DNA comes in, the protein's taken off, the RNA is taken off, and it is repaired. The gap is filled in by a DNA polymerase. It's not DNA replication, that is DNA repair. So it's a really important distinction. So that Adenovirus genome is made double-stranded, and now it can be a template for transcription. The second one there, parvovirus. These are small DNA viruses with single-stranded genomes, and they have a little interesting T structure at each end. They're not ready for transcription because only double-stranded DNA is ready. So this has to be repaired. So DNA, the cell fills that gap in there, but it's not replication. We're not making more genomes. And I'm telling you this because I already told you that the first thing that happens when a DNA virus gets into a cell is you got to make a protein. But you can't do that until you repair these genomes, but it's not technically DNA replication. This is not DNA replication. And finally, the retroviruses, the RNA is in the particle as a plus strand, but it's not released from the particle. That's why it's not translated. It is instead copied into DNA by the reverse transcriptase, which is in the particle. And that DNA then integrates into the cellular genome. It becomes what we call a provirus, and there it is transcribed by cellular polymerase. So that's how these genomes are fixed in order to be ready for transcription. Which viral genomes do not need conversion? The double-stranded ones. SV40, the polyomaviruses, papillomas, adenoviruses, herpesviruses. And so they're all double-stranded. They can go right in and be transcribed. Anyway, so that's here. The viruses that are double-stranded DNA can go right to mRNA, adenovirus, herpes, polyoma, and papilloma viruses. So let's talk about this process today, transcription, DNA to mRNA. It's a very specific term. It's not translation, it's not protein synthesis, it's not RNA to RNA. The process we talked about last time is not transcription, it is RNA synthesis. This is transcription from DNA. Most viruses use host cell RNA polymerase to make mRNA. The exceptions are viruses that replicate in the cytoplasm, like pox viruses and some of the giant viruses. They do everything in the cytosol, so they have to encode their own RNA polymerase because all the other viruses go in the nucleus where RNA polymerase is, because in our cells, we're copying our genome to make mRNA that happens in the nucleus. So mRNA synthesis is done by RNA polymerase II, but there are 3 enzymes in the cell that make RNA from DNA templates. There's, of course, Pol1, which makes ribosomal RNA. As far as we know, no viral RNA is produced using Pol1. Pol2 is utilized by many viral genomes. Here on the right are the viral kinds of RNA that are made from Pol2, messenger RNA or pre-messenger RNA, the precursor. Primary microRNAs, the hepatitis delta virus genome is actually copied by RNA Pol2. We'll talk about that separately, and there's some others, Herpesvirus 8, polyadenylated nuclear RNA. But the main one, of course, is pre-mRNA. For cells, we have our pre-mRNA made by Pol2, our microRNAs, small nuclear RNAs, and long non-coding RNAs. That's Pol2. And of course, Pol3 makes mainly tRNAs, ribosomal RNAs, and small nuclear RNAs. And these actually participate in splicing. Some viral genes are transcribed by Pol3. They include short RNAs like adeno2 VA RNA. We'll talk about that function later on, and a couple of others. These are 3 different polymerases. They all initiate synthesis de novo. They do not need a primer. As far as we know, no DNA-dependent RNA polymerase needs a primer. And so that's a lot like some of the RNA polymerases that we talked about last time. So this is an overview of the transcription and processing scheme, which applies to cellular RNAs as well as viral. We'll go over it. It mostly occurs in the nucleus, which is at the top. There's a double-stranded DNA genome, could be viral or cellular. It has a promoter at one end, which we always show as a red arrow, and that's where transcription begins. Shortly after transcription begins, the RNAs are capped. A G-PPPN is put on, so instead of a 5 to 3 prime base-to-base linkage, it's a 5 to 5 prime. It's special. So that's put on very early in transcription. Then we have production of a pre-mRNA, a precursor. It's a precursor because it has sequences that are going to be removed in the mature mRNA. This precursor is cleaved at a polyadenylation site indicated by the arrow. It's then polyadenylated by a set of enzymes, very different from how it worked for RNA viruses. These precursors often contain introns, intervening sequences that are removed by splicing. And then the mRNAs are exported into the cytosol where they can be translated. Most of our mRNAs are spliced. Many viral mRNAs are spliced as well. In general, the process of splicing marks the mRNA for export out of the nucleus. It's marked by proteins being put on it during the splicing process. However, there are many viral genomes or viral mRNAs that need to get out without being spliced. And so there are special mechanisms for that, which we'll talk about later, so that some unspliced genomes can get out. And they can be translated as well or put into virus particles. Now, an important aspect of this, of course, is mRNAs don't last forever. Some of them have very short lives, a few seconds, 30 seconds, a minute. Some of them last hours. And eventually they're all degraded so that you can control gene expression. Let's talk a little bit about the initiation of transcription, because it's highly regulated in us, but also in virus-infected cells. And here we're looking at a transcriptional control region of a typical DNA. And here's our promoter on the right, where mRNA synthesis begins. That is surrounded by what we call an initiator sequence, which specifies the polymerase to come and start making mRNA, specifies accurate starts. A little bit upstream, 20 to 35 base pairs upstream, is a TATA sequence. That's the sequence of the DNA, TATAA, and that binds a particular transcription factor called TF2D, which is part of the whole complex that assembles the polymerase on the DNA. And these two constitute what we call the core promoter. You could take those and put them in front of any gene and get transcription in a cell. And we have some other upstream sequences. We have local regulatory sequences that bind transcription proteins and allow for regulation. And then we have distant regulatory sequences. They can be, 100 to 10,000 bases upstream of the actual promoter. And these include enhancers, which stimulate transcription, and silencers, which repress transcription. So all of these are sequences involved in the regulation of transcription, and many of them function by binding proteins which themselves modulate the process. And we know most of this from analysis of viral genes, but we know now that we have similar structures in cellular DNAs as well. So here are just 3 examples to show you transcriptional control regions of 3 different viral promoter regions. So on the top we have what we call the adenovirus type 2. Adenovirus occurs in many serotypes. So that means if you're infected with serotype 2, you'll be immune to serotype 2 next time you're infected, but you could get infected with any of the other serotypes. Makes vaccine synthesis challenging. Anyway, that's a major late promoter of AD2. There's the promoter at +1, and you can see upstream a number of transcriptional regulatory sequences that bind specific proteins like CPF and USF1. And these regulate transcription. Here's the SV40 early, a couple of transcription start sites here at the promoter area, and then upstream, again, sequences that bind specifically proteins, in this case SP1, repeated over and over. And down here is the adeno early promoter, E2 early promoter. There are a number of different adenovirus promoters in the genome. And in fact, some viral genomes can have 1, 2, or more. And here again, upstream binding sites for proteins. Again, these are DNA sequences that specifically bind proteins to help regulate the process. Transcription is highly regulated. You don't need the same amount of mRNA in every cell type in our body at the same time in development. And in the same way, viruses do not make all the mRNAs at the same time during infection. It has to be regulated. And in part, these proteins play a role in that. And viruses can make various proteins to regulate this process. The actual step of transcription initiation and elongations are shown here. We know a lot about this now. We know structures of the polymerase and many of the proteins that are binding with it. So here at the top is our promoter region with the promoter start site. And all of these proteins, there's the RNA polymerase 2 on the right, but all these other proteins need to come together and assemble an initiation complex. And some of them bind to specific sequences in the promoter region, like TF2D, and others bind each other. But in the end, and they're all called initiation proteins, you have what's called a closed initiation complex. A little bit of energy is needed in the form of ATP to open it up so that it can start transcribing. And the first few cycles of transcription where it's copying the DNA template, you see these little green lines, those are mRNAs. It undergoes abortive transcription until a minute or a couple of minutes, it finally gets going into transcribing the whole DNA. And we think that this abortive transcription is to ensure there's no primer here. So we think that the polymerase isn't really sitting down accurately on the DNA without a primer. So it does a few rounds of synthesis, which we call abortive synthesis, before it really gets going. DNA polymerases don't do that. They start right away because they have a primer, as we'll see next time. So as this transcription begins, once it gets going past 30 or 40 bases, then the cap is put on. So you don't bother to cap the abortive transcripts. That would be a waste. Capping happens as the RNA polymerase moves downstream. So again, this happens in virus-infected cells. This enzyme is, for the most part, a cellular enzyme on all the DNA viruses except those that replicate in the cytoplasm. One last aspect of this initiation process is how an enhancer works. So these can be up to 10,000 base pairs away from the promoter. And the way they work is they bind proteins which loop the DNA around, and those proteins interact with proteins that are already sitting down at the promoter. They help to stabilize the transcription complex, the initiation complex, and they stimulate transcription. You take them out and transcription levels go down. Similarly, there are also silencing sequences that bind proteins which will silence the transcription. So lots of ways to get regulation of gene expression. We've been talking a lot about proteins involved in this process besides RNA polymerase II. A lot of other proteins are involved, and there are two kinds of proteins here. First, there are sequence-specific DNA-binding proteins. So all those colored regions on the DNA that I was showing you that have funny names that bind proteins, those are sequence-specific DNA-binding proteins. So only the right protein will bind to that sequence, and it won't bind anywhere else, and that protein has a certain function in transcription. We also have what are called coactivating molecules. These do not bind DNA, but they are part of the initiation complex, and they can modulate transcription in a variety of ways. And one of them is by modulating the structure and the activity of nucleosomal templates. So what we were talking about before, the pattern of methylation and acetylation on the histones can control gene expression. And these coactivators can modulate that. And we'll see an example of that today. And the idea is that the methylation pattern of the histones, the acetylation pattern, can affect initiation, either by loosening or tightening up the DNA wrapped around the histones, for example. So two kinds of proteins, sequence-specific binding proteins and coactivators that don't bind DNA but still regulate transcription. We have these in our cells, and some viruses encode their own to modulate the activity of the host cell RNA polymerase. Here's how a sequence-specific DNA binding protein is built. It's typically modular, and 3 domains are shown here. You can actually take these and mix and match them and get different specificities and activities of different proteins. At the N-terminus, we have a DNA binding domain. So this is the part that attaches to the DNA. Remember, it's a sequence-specific DNA binding protein, and that's encoded in that N-terminus. These have different motifs like zinc fingers, helix turn helices, basic motifs, all different names for this DNA binding region. Then there's a region that's typically a leucine zipper, a stretch of leucine residues which allow 2 monomers to form a dimer. These proteins typically bind DNA as dimers. There's a nuclear localization signal. Of course, these are made in the cytoplasm. They have to get into the nucleus. And then at the C-terminus, an activation domain, which has a variety of features. It can be acidic, glutamine-rich, proline-rich, isoleucine-rich. They're all different kinds. But these do the business end. They interact with other proteins to stimulate transcription. So that's how these are organized. And you can, as I said, move these around among different proteins to get proteins of different activities. Our first question is, what is the first biosynthetic event that occurs in cells infected with double-stranded DNA viruses? So with membrane fusion, transcription, DNA replication, protein synthesis, all of the above. See how we did? Most of you got transcription. That's right. That's the first biosynthetic event. It's not DNA replication. That is a biosynthetic event, but it doesn't happen. Only DNA synthesis, yes?
So all the transcription factors that viruses use for their regulatory regions are the cells, right? Because they don't encode their own. Or do they?
Some do. We're going to see an example of that today. But has to be after the initial wave of transcription, right? So they can encode proteins that'll change the pattern, but the first one always has to be made using the cell machinery. Exactly. Yes, so they use the same DNA binding proteins that the cell uses, and that's what they have to do to start, absolutely. Yeah, but membrane fusion is not a biosynthetic event.
All right.
Right? It's just two membranes fusing together, yeah. So membrane fusion isn't not DNA replication. It's not protein synthesis, it's transcription, making of mRNA. I want you to get this. That's why I'm making a big deal out of it, because I think conceptually it helps you. And that question was great because, well, doesn't it have to have our transcription factors? No, the first event will always use the cell unless there's a transcription factor in a virus particle. And we'll see an example of that today. That can happen. So here are the different strategies for transcription of viral DNAs, from simple to complicated. We're going to go through some of these today. On the left, we have the origin of the transcriptional components, and on the right, some virus examples. First, we have host only. These viruses, retroviruses with simple genomes, they use the entire transcriptional system, as we were just saying. Polymerase are plant retroviruses. Circoviruses, the smallest DNA viruses. They do everything with the host. They make nothing of their own for transcription. Again, for DNA synthesis, they have to make at least one protein, but not for transcription. Then we have host plus one viral protein, and there are two different examples here. First, the viral protein transcribes late genes. And there are two examples in phages where it initially is transcribed by the host polymerase, but then the virus makes its own RNA polymerase. Host plus one viral protein, then we have a viral protein. So the virus is using the host polymerase, but it makes one viral protein that regulates transcription. And that includes hepatitis B, parvo, papilloma, polyoma, retroviruses with complex genomes. We'll see an example of this today. So it's one viral protein that regulates transcription by the host RNA polymerase. And then moving to more complicated, the host components plus more than one viral protein to stimulate transcription. Adenoviruses and herpesviruses, which use two or three, as we'll see, or more. And then all viral, all of the transcription machinery, polymerase, host proteins that bind DNA, it's all viral. Those are the viruses that reproduce in the cytoplasm of the cell, the pox viruses and the giant viruses like viruses. Because they can't access any of the transcription factors because they're in the nucleus. They can't access RNA polymerase because it's in the nucleus. So this makes sense, actually, if you think about it, because to replicate exclusively in the cytosol, you need to encode everything yourself. So from very simple, where only host components are used, and not even a single protein is made to modulate host transcription, to intermediate, all the way to all viral. So we'll look at some examples of this today. But first I want to tell you some paradigms about how we view these transcriptional systems. And what I want to tell you about today is how it's regulated in viral infections, because as I said earlier, not every mRNA is made all at once. Has to be regulated, and why it needs to be regulated will become evident in a bit. On the top we have what we call cascade regulation, which you will see specific examples of. Here, the DNA of the virus gets into the cell. The first gene here, gene X, has a promoter that's recognized by the cellular machinery. It's transcribed by RNA polymerase. You get an mRNA, and you get a viral protein produced. Then that protein is needed for transcription at a second promoter. That second promoter, which is in front of gene Y, it will not work without protein X. That's called cascade regulation. And you will see increasing complexities of it where we have two component like here and even three-component cascade regulation. It's a way of regulating what proteins are made. And I'll give you a hint. When a virus first comes in this cell, a DNA virus, there's no point in making structural proteins if there's no DNA to put in them. So the structural proteins are always delayed until late in the reproductive cycle. All right, that's cascade regulation. Then we have an autoregulatory loop, which can be positive or negative. And here we have an example of a positive one. We have a viral DNA with a promoter, and it's upstream of gene X. This comes into the cell, it's transcribed by cellular proteins, you get an mRNA, you get a protein made, and that protein increases transcription from promoter of gene X. So at the onset, the promoter is at a basal level. As soon as some protein is made, it increases. So you get more and more and more transcription and production of cellular protein. So that's a positive loop. We also see negative loops where proteins that are made can inhibit the transcription at a promoter. Some proteins can do both. They can activate one promoter and inhibit another as a way of making a cascade regulation. But these are the two essential elements to controlling gene expression: cascade regulation and autoregulatory loops, and they're accomplished by proteins that participate in the transcription process. So we're going to go through three DNA viruses, and look at their transcriptional programs and see how these principles work. And this is the first, the simplest. It is SV40. It's a polyomavirus. These are small DNA viruses, circular double-stranded DNA in the particle, and it's chromatinized, as I pointed out earlier. Here's the genome on the bottom there. It's relatively simple. And there is this ORI, it means origin of replication. That's where DNA synthesis occurs. We'll look at that next time. But at the ORI, there are also two promoters going in opposite directions. And that's shown by the green arrows. There's an early promoter and a late promoter. So this is already telling you what's going on. There's some temporal regulation. When this virus infects the cell, only the early transcription occurs, not the late, and a protein is made from the early, and then the late transcription occurs. And that's shown on this timeline here. So we have an early mRNA made. So that promoter, the early promoter, is recognized by the cell machinery. It's made into an mRNA that makes this protein LT, large T antigen. And that large T antigen will stimulate DNA replication and the late promoter activation. So this is an example of a protein needed for the late promoter to work. It's made early in infection. It turns on late promoter, it turns on DNA synthesis. As we'll see, it also participates in DNA replication and is an oncoprotein which can cause tumors. So this is the very simple pattern. Early phase and late phase. This purple bar always means the onset of viral DNA synthesis. So in the early phase, there's no DNA synthesis. In fact, that's how early and late are typically defined. Late is always at the onset of DNA synthesis. So the early promoter is on first, makes LT mRNAs. LT is made, and they can turn on DNA synthesis, and they will also turn on the late promoter. There's also a role for DNA synthesis of anti-repression of the late promoter, which I will show you right here. So when DNA molecule gets in the cell, the SV40 DNA, the late promoter is not active. One reason is there's no large T, but another reason is there's a cell protein that's binding the late promoter that's repressing it. And that's called the cellular repressor IBP, initiator binding protein. It's a yellow protein, it's a cell protein. And when the virus DNA first gets in the cell, it's coated with this yellow protein. At least the late promoter control elements are coated with it. So here on the right is the late promoter, and these pink bars, these pink boxes are transcriptional control regions, and the initiator binding protein, a host protein, binds to those and represses late transcription. So clearly the virus has evolved to utilize this mechanism of late repression. Early promoter works because the IBP does not bind those control sequences. On the left is a graph showing you the time after infection with the concentration of viral genomes in blue. So the early phase, there's no DNA synthesis, and then the late phase is defined by DNA synthesis. The viral late RNA is in green. You can see there's no late RNA early because it's repressed by initiator binding protein whose level stays constant throughout infection. It's a cell protein that doesn't change in response to infection. So that's one mechanism of repressing the late promoter. So how does it get turned on? Well, when there's enough T antigen produced to turn on DNA replication, you start to get multiple copies of the viral genome made. There's a fixed amount of initiator binding protein, so it's titrated out, basically. There aren't enough proteins to bind all the DNA molecules that are made, so eventually you have DNAs that are not bound, the inhibitory proteins, and they can start transcription of the late genes that will get DNA replication going. All right, so that is how you make two phases, and you make the second phase dependent on DNA replication by having it titrate out this IBP. And why? Again, because you don't want to make late proteins until there's DNA to go in it. So there's no late proteins made early by definition, so you make DNA. And then these mRNAs also encode the structural proteins. So here's an overview of the replicative cycle of this virus. The viral DNA is in the nucleus. It is immediately transcribed to an mRNA, which is exported. It encodes large T. Large T goes back in the nucleus. It stimulates transcription and eventually DNA replication. Once DNA replication begins, the late promoter is de-repressed. So now you have late RNAs transcribed. That's shown here by this dotted line, the early and the late phase. Those late mRNAs go out, they're translated into structural proteins. Those have to go back in the nucleus because DNA replication is occurring in the nucleus, and new virus particles are assembled in the nucleus. And so here again, the function of early and late phase is to delay synthesis of structural proteins until DNA has been replicated. And so the late promoter is off until DNA starts replicating. It's then on, and then you get viral proteins made. It's a wonderful connection between the two. Now, a little more complicated adenovirus, where we have three phases now. We have an immediate early, and a late phase. And here we have three viral proteins controlling this, in addition to DNA synthesis, as we saw before. We have the viral DNA comes in. The first protein made is E1A, early region 1A protein. And that's needed for all transcription of the early genes. Okay, so you don't do anything until you make E1A. You'll see why in a moment. Then E is made. The E2 proteins are needed for DNA synthesis. DNA synthesis stimulates entry into the L phase. You then get transcription at a late promoter, which encodes the structural proteins. All right, so these four proteins, E1A, E2, and these two are needed for enhancement of late gene transcription. A little more complicated, but it's a bigger virus, as you will see. The need for E1A, for an immediate early protein, is because the virus requires a cell transcription factor that is normally not available. So remember, as we said before, this virus DNA that comes in has to be transcribed by the whole cellular machinery, and that makes the E1A protein. And then E1A's role is to free up this transcription protein called E2F. E2F is needed for the transcription of the viral early genes. Normally, this protein, this is a cell transcription protein. It's bound to a protein called RB. RB stands for retinoblastoma. It was discovered in tumors of the eye. We're going to come back to this later in what is a fascinating story. But for now, most of the cells in our body are quiescent. They are not dividing. And that is because E2F is blocked by RB. And the genes that are needed for cell division require E2F. And so this is a way to keep the cells from dividing unless it's necessary. So here's a promoter in the cell. And here we have E2F. It happens to be in with another protein called DP1, but it's not important for us. It's bound to RB. This promoter is repressed because this complex recruits histone deacetylases to the promoter. Now, histone deacetylases take acetyl groups off of chromatin and makes it tighter, and it doesn't transcribe. So acetylated histones are actively transcribed because they're loose, the chromatin is loose. Histone deacetylase removes the acetyl, closes up the chromatin, inhibits transcription. So RB recruits H stacks to whatever promoter it's bound near, and that shuts it down. However, adenovirus requires E2F, so the E1A protein binds RB, knocks it off of E2F, and now the virus can use E2F, and of course the cell will start dividing. And the virus will also need that for DNA synthesis, as we'll see next time, so this has a dual function. Again, the virus requires E2F to transcribe the early genes, the E right here, and that's the function of E1A to free up E2F, which is a family of cellular transcription proteins, so that the viral early genes can be transcribed. Here's a map of the viral genome, a transcription map. Long double-stranded DNA, about 40,000 bases. Here you can see the transcription occurs from both strands, so there are promoters on both strands, so it's complicated. That's why it needs to be regulated in 3 phases. The early genes are here on the left. E1A is the one we just talked about, is freeing up E2F. That promoter is operative first. And then there are E promoters on the other strand, that second wave of mRNA synthesis, the early phase. And you see they include DNA binding protein and the DNA polymerase. So that's going to turn on DNA replication. And once you get DNA replication, the late promoters are activated, and they're on the top strand. Here is the major late promoter, which encodes a huge mRNA precursor that encodes structural proteins. Look, penton base, core proteins, hexon, etc., fiber. 3 waves of transcription, nicely regulated, so that DNA synthesis coincides with the synthesis of structural proteins. It's just beautiful, isn't it? All right, you need to think about it a long time before you think it's beautiful, I guess. I've had a long time. All right, so here's the replicative cycle. I agree. My first 10 years, I didn't think it was beautiful. But then I started to, actually what made it beautiful to me was writing a textbook that put all of it together, okay? Because here's the secret, most virologists, most scientists only think about what they work on, pretty much. But since I wrote a book, I thought about pretty much all viruses, and this unification made me appreciate the totality of these individual viruses. So here on a replicative cycle, we're going to look at immediate, early, and the late phases. We've talked about adenovirus getting into cells. The partially disassembled capsid docks on the nuclear pore. The DNA gets inside. So that is immediately recognized, and the E1A mRNA is made. And that goes out into the cytosol. E1A protein is made. It comes back in the nucleus. It binds RB, frees up E2F, and now you can get early transcription. And that includes the genes. So these are the early mRNAs there, the second set. They are translated into the DNA polymerase and all the proteins you need for DNA replication. So those go back in the nucleus. They start replicating the genome, and that replication alleviates the repression of the late promoter. So you get late genes transcribed, and those encode structural proteins, which again go back in. And since DNA replication is occurring in the nucleus, that's where you assemble the particles. So you ship the structural proteins back in. So again, starting with the promoter that's recognized by the host cell machinery, you then make proteins that can expand the transcriptional activities. And finally, the last transcriptional program is Herpesvirus. So the adenogenome was about 40 kb long, and these are longer. They can be 140 kb or longer, 140,000 bases or longer. Here we have immediate, early, and late, similar phases of replication, but we have a twist. And that is this protein, VP16, which is actually in the virus particle. So there's a herpes virus particle. It has a membrane envelope, and in it is a nucleocapsid, an icosahedral shell with the DNA inside. But in between the capsid and the membrane, there are many, many proteins of various sorts, and one of them is a protein called VP16. VP16 is needed to allow the first promoter to work. Otherwise, it doesn't work. So this is an exception to what I've told you so far, that the viral DNA needs to be recognized by the host machinery. This will not be recognized. The immediate early promoter is not activated until VP16 comes in with the particle. How it gets there, I'll show you in a moment. So this is different from polyomaviruses and adenovirus because their early and immediate early promoters work in the cell without any other viral proteins. This one does not. It needs this protein VP16. All right, so here this activates immediate early transcription. The promoter is poorly recognized by the cell. So then you make immediate early genes, including ICP0, which will stimulate the production of the early genes. The early genes encode the DNA replication machinery. That will get DNA replication going. You relieve the repression of the late promoter, and then you get the late genes that encode the structural proteins. Similar situation to the other two viruses with some nuances. Again, delaying synthesis of late genes, the structural proteins, until you get DNA replication, and having a way to coordinately express all these proteins. But this is unique. This VP16 is really interesting because we don't see it in those other viruses. So here's the replication cycle. The virus is binding. It fuses at the plasma membrane. And all those proteins that are around the capsid spill out into the cytosol. And VP16 has a nuclear localization signal. It gets into the nucleus. It's a protein getting in the nucleus. At the same time, the capsid is moving to the nuclear pore. The DNA comes out of the portal. It's in the nucleus. That immediate early promoter is activated by VP16. You get immediate early proteins. They go in the nucleus, and they activate the early proteins, which encode DNA replication proteins. They go back in, the DNA replicates, and now you get the late proteins, the late mRNAs produced, and they encode the structural proteins, which come in and help assemble the newly synthesized capsids. All right, so, and again, this shows you how this viral DNA, when it comes in the nucleus, it's immediately wrapped around nucleosomes. The cell attempts to silence it, actually, because it doesn't like to have foreign DNA coming in. And the virus encodes proteins that reverse the silencing. Otherwise, it wouldn't be transcribed. Why does the DNA circularize? Because the way the DNA replicates is by rolling circle. I don't know if you know that mechanism, but phage lambda does that. It makes a circle, and then you start copying this, and the circle rolls, and you get a very, very long concatemer of genome after genome after genome. Those get put into the capsids and cut. We'll see that next time, actually. Concatemeric DNA. Yeah. Next. All right. Adeno E1A stimulating expression of E2, which then stimulates adeno 4A and L4 is an example of. So it's early, immediate early, late is an example of negative autoregulatory loop, repression of gene expression, cascade regulation, dimerization. All right, how did we do? Cascade regulation, absolutely. Most of you got that. It is not a negative loop. Part of it may be construed as that, but it's certainly this cascade, you know, early, immediate early, late, that is cascade regulation. All three of those viruses. A few words on the RNA that's made, mRNA. The 5' end is a cap structure that's put on shortly after initiation begins in the nucleus. This is what it actually looked like. I've told you about it a few times, but that first base is added in a 5 to 5' linkage. There's the 5' end of the base. There's the 5' end of another. Normally it's 3', 5', 3', 5', but here it's 5', and you have all three phosphates on it as well, as opposed to one. So that's called a cap structure. Has a variety of functions, including enhancing translation, mRNA stability, and transport, and so forth. So that's capping. And the other thing that happens is you have polyadenylation, and I just want to go over this briefly because it's very different from the mechanism we talked about last time. Here we make a precursor which is cleaved at a specific site, and then poly-A is added by an enzyme. That happens in the nucleus. It's shown here. Here's our pre-mRNA. There is a poly-A addition site, AAUAAA, which is recognized by a protein. It is cleaved, and then poly-A is added by one of two poly-A polymerases, up to about 200 bases. So the transcription goes beyond the poly-A site, and then it's subsequently cleaved and polyadenylated by separate enzymes, so that all the mRNAs coming out are capped and polyadenylated. And so we can now compare this with last time, mechanisms, enzymes, and viruses of adding poly-A. We have post-transcriptional, which is the one we just talked about, cleavage of mRNA followed by polyadenylation, and that's done by cellular enzymes, and all these viruses that we've talked about today do that. And then the other kind of polyadenylation we talked about Monday, which is during mRNA synthesis. We talked about reiterative copying at U stretches in the viral RNA for flu and VSV, or copying of a U stretch in the viral RNA, polio, alpha viruses. The A is present on the RNA. It's copied into U's when you make the complementary strand, and then it's copied back into A's, so it's encoded. I want to talk briefly about splicing and how it contributes to viral replication. Splicing of mRNA, which now we know happens with all eukaryotic mRNAs, was discovered in cells infected with adenoviruses. And the discovery arose because when people were extracting RNA, viral RNA from the nucleus in virus-infected cells, they found it was always bigger than the final mRNAs in the cytoplasm. And a key experiment, they took these mRNAs from the cytoplasm, the mature mRNAs, and they hybridized them to single-stranded adenovirus DNA. And they looked at these in the electron microscope, and they saw this picture here, which is a tracing of the original heteroduplex experiment, but I've drawn it for you here to simplify. What they saw was the DNA was longer than the mRNA. There were these loops coming out. And we now know that these loops represent introns that are removed from the pre-mRNA by splicing. And so this turned out to be the gene for the hexon protein. And the genome looks like this. It encodes, the body of the protein is encoded down here in green, but the rest of the RNA, most of it is spliced out. The red, the orange, and the blue parts, red, orange, and blue, are spliced out, and only 3 exonic sequences remain on the hexon mRNA body, which is the green mRNA there. And that discovery got the Nobel Prize in 1993 to Richard Roberts and Phil Sharp for this discovery, which went on to be found in every virus and in every cellular mRNA. Really a remarkable discovery. And one of the reasons it's powerful is that you can get a single pre-mRNA spliced in different ways and get different proteins made as a consequence. So for example, here are some ways that can happen. On the left, we have our pre-mRNA with 3 exons and 2 introns. The introns in pink, they're going to be removed. This constitutive splicing, we remove both introns. You get 1, 2, 3 exons put together. You can have alternative splicing, where the splicing jumps over one of the exons. So you can have 1, 2, 3, or 1, 3, for example. Sometimes there are alternative splice sites. There can be alternative 5' and 3' splice sites. So for example, here on the top in the third from the left, you have 5A and 5B, which can be used to splice to the site before intron 2, and gives you 2 different mRNAs. And the same way you can have alternative 3' splice sites. So this gives you a lot of coding capacity in a genome. And I want to show you an example of that in the adenovirus genome, just how amazing this can be. This is the late mRNA, the major late mRNA, which is transcribed from the top strand, the major late promoter. It's not activated until DNA synthesis occurs, but it encodes all the structural proteins. So you make a very long mRNA, and then there are 5 polyadenylation sites. 1, 2, 3, 4, 5. So it can be cleaved at any one of those 5 sites, and poly A can be put on. And then here you see the mRNAs that would result from the polyadenylation first. They have still introns in them. And then you have alternative splicing in a variety of ways. You have different 3' splice sites on the left, different ones on the right, and you end up with multiple mRNAs in both cases. So many different mRNAs encoding various proteins. Here's the P4 structural protein, the hexon, the protease, and so forth. So you can make lots of different proteins from a single transcriptional unit. So you can imagine that in a virus with a rather small genome, that can be powerful. And SV40 does splicing like crazy to get a lot of proteins out of its small genome. All right, our last question today. Which statement about polyadenylation of DNA virus mRNAs is correct? It always occurs in the cytoplasm. It occurs after cleavage of pre-mRNA. Poly A is added at the 5' end of pre-mRNA or is specified by a stretch of U residues in the template. DNA virus mRNAs. All right, let's see how we did. It occurs after cleavage of pre-mRNA. That's the right answer. Not all of you got it. So the polyadenylation doesn't occur in the cytoplasm. It happens in the nucleus. It's not, poly A is not added at the 5' end. It's at the 3' end. The cap is at the 5' end. Stretch of U residues in the template. Those are RNA viruses that do that. This is a DNA virus where there's just a cleavage site and then an enzyme adds poly A. The process of splicing marks mRNAs for nuclear export. There are a whole series of proteins that bind the pre-mRNA during splicing. So here's a pre-mRNA with an intron, a lot of proteins bound. The intron's removed. This is an autocatalytic reaction, by the way, an RNA-based reaction, probably a remnant of the RNA world. And these proteins are then recognized by the nuclear export machinery to get the RNA into the cytosol. And so that's why most of our cellular mRNAs need to be spliced in order to get out of the nucleus, because they're marked by these proteins recognized by the export apparatus. But as I said earlier, some viral genomes need to get out unspliced. So some retroviruses, their genome is packaged in the virus particle as an mRNA. It has to come out unspliced. So how does that happen? Here's an unspliced retroviral RNA. If it's not spliced, it's not going to be marked with these proteins. Well, this happens to have a little sequence at the 3' end of the RNA called CTE, constitutive transport element. It binds the proteins that are recognized by the export machinery, and that's how it gets exported without splicing. It's brilliant. So this genome sometimes is spliced, and the spliced products makes proteins, and they get out without a problem, but it has to be unspliced because that's going to be the viral genome, and the CTE does that. So that's one example. I want to show you one more for HIV, where the same idea is the viral genome is transcribed as a provirus into a long mRNA, and that's spliced to give different mRNAs that are exported because they're marked for export. But you also have to get the full-length genome out to be packaged into new virus particles. So how does that happen? Well, there's a little sequence at the 3' end called an RRE, a Rev response element, and one of the proteins that are made by spliced mRNAs, Rev, comes back in the nucleus, binds this long mRNA, and that signals the export machinery to take it out. So clever ways to get around this requirement. And finally, splicing is value added. Basically, I've shown you all the different ways you can get various mRNAs from a precursor. You can make different mRNAs or proteins, so you expand the coding capacity of a small DNA genome. And what I haven't shown you is you can regulate gene expression by modulating splicing. We won't go into that, but you can control how this splicing works by various viral proteins. So you might need a protein at a certain part of the cycle, you can control it. So far we've talked about messenger RNAs. As you know, there are non-coding RNAs in the cell, like tRNAs and ribosomal RNAs, but there are other ones that have been discovered in the past 10 years as we have the capacity to sequence all the RNAs in a cell, total RNA-seq. We've found that, in fact, most human transcripts don't encode protein. They're mostly non-coding. And these have been classified into short non-coding RNAs, which are 200 bases, and long, which are greater than 200. They have a variety of regulatory functions. Not only do cells encode these, but viral genomes do as well. And here are some of them on the right. There are microRNAs, short RNAs that regulate gene expression. There are long non-coding RNAs that also regulate gene expression. And the latest discovery, circular RNAs, which are also regulatory. So let's look at some of these. A very famous kind of non-coding RNA is microRNA. These are made from Pol2 enzymes. They make primary microRNAs, which are then processed into short hairpins, as you can see here, by an enzyme called DROSHA. So now we have this little pre-microRNA, which is exported. It's further processed by dicer. It associates with a protein called argonaute. One strand is removed, and now in this complex, it can silence genes. And here are two examples. Here's a mRNA being translated. Here's the cap. Here are ribosomes translating the RNA. So this microRNA, these microRNAs are specific for sequences on mRNAs. They're about 21 nucleotides long. They base pair with the mRNA. They can do one of two things. They can suppress translation of the mRNA, or they can degrade it. Argonaute is a nuclease that will degrade the target mRNA. So these little microRNAs can regulate gene expression. We know that there are cellular microRNAs that regulate viral gene expression, and there are some viruses that encode microRNAs that regulate both cellular and viral gene expression. So that's one kind of non-coding RNA. It's a really interesting microRNA in the liver. It's called miR-122. And it's required for the replication of hepatitis C virus. This is a virus that causes hepatitis in many people. We'll talk about it later. It's a plus-stranded RNA genome. It's a flavivirus. At the 5' end is a highly structured region expanded at the left here. And at the very 5' end, that little stem loop, there's a binding site for two miR-122s. There's one in red and the second in red. If you don't have the miR in a cell, the virus RNA is degraded and it doesn't replicate. And this miR-122 is liver specific. And that's why this virus is hepatotropic. It can't replicate in other cells because there's no miR-122. If you put this artificially into another cell type, you can get the virus to replicate. So that's a very cool way that a virus has evolved to exploit a microRNA. Here's another example, polyomaviruses that we've been talking about today, SV40. It's a late transcript processed into a microRNA that has targets on the early mRNA. So there's the early and the late promoters. The late mRNA encodes this pre-microRNA, which targets and degrades early mRNAs. And we're not sure what the function of this is, but there's some idea that this allows the virus to persist in the host by preventing DNA replication and packaging of new viruses. There are many, many long non-coding RNAs in cells, and we're just beginning to understand what they do. They're long RNAs, longer than 200 bases. They don't encode any protein. There's some examples here. Here's the cytosol. There's one encoded by this ACOD1 gene that reshapes the metabolic environment of the cell and increases the replication of Sendai virus and vesicular stomatitis virus. We'll look at that later. And then there are many long non-coding RNAs that act in the nucleus. And here are three examples where they're binding and recruiting transcription proteins to regulate transcription. And so for here, this particular one, NRAV, it is downregulating a number of antiviral genes. So influenza A virus replicates better. The virus has evolved to utilize these LNC RNAs to replicate better. There's another one that affects the transcription of interferon gamma, downregulates interferon gamma, which is needed for virus clearance. It increases the persistence of a virus, Tyler's murine encephalomyelitis virus. And then There is another one that increases the transcription of genes involved in antiviral responses, and that reduces the replication. So these long non-coding RNAs have lots of positive and negative effects on virus reproduction. Really quite interesting. And this is really early days in the research on these. It's just beginning to be sorted out. And precisely how they work is an interesting question. In this case, for ACOD1, it binds an enzyme in the Krebs cycle and can activate it, and that reshapes the metabolic activity of the cell and allows the viruses to replicate. So quite interesting ways of working. And finally, circular RNAs have recently been discovered both in uninfected and virus-infected cells, thousands and thousands of different ones. They're made by back splicing. So here are some introns in a pre-mRNA, 1, 2, 3, and 4. And normally you would splice in a 5 to 3' direction, but sometimes you back splice from 3 to 5'. That makes a circle, as you can see here. These have introns that they can be coding or non-coding. And we don't really know what the function of these are. We find lots of them in tumors, so people are thinking about using them as tumor markers. But there are two activities of these that have been discovered, shown on the right. They can be sponges for microRNAs. So microRNAs, remember, target mRNAs and degrade them or inhibit their translation. We think some circular RNAs can be sponges. They're hanging on it inactive. And at some signal, they would come off and go to the target. And we also think they can hang up RNA binding proteins that are involved in, say, splicing or other RNA metabolism. So these circular RNAs, really interesting, really early days. We don't quite know what they do, but lots of work going on. It's something just recently discovered, process, a byproduct of splicing. All right, the last thing I want to tell you is RNAs can be modified. We've only learned about this in its functional aspects in the last few years, and one of them is reversible N6 methylation of adenosine nucleotides. So here's an adenosine. The N6 position can be methylated. There are enzymes that will methylate that position. Those are above the line. There are enzymes that will remove the methyl, and there are proteins that sense the presence of a methyl. We call these writers, readers, and erasers of the methyl groups. And the methyl groups have effects on the function of the RNAs. They can inhibit viral RNA production, or they can stimulate it. And here's one example where we're looking at the effect of methylation on hepatitis C virus mRNA. So here's the viral RNA. Normally it gets encapsulated on lipid droplets to make a virus particle. But if it's methylated, the RNA cannot be incorporated into the lipid droplet, so virus yield is reduced. So here's an example of where methylation is a cell defense against the virus. So it's put on by an enzyme, and then another protein can read it to sequester it. Very interesting, and we may come back to that. So we've talked about transcription of DNA genomes today. Remember, key point, DNA has to be double-stranded. If not, it first has to be fixed before it's transcribed. And then there are typically cascades of regulation of transcription in infected cells. So next time, we're going to talk about how these diverse DNA molecules replicate during infection.